So thank you, everyone, and thank you especially for the organizer for inviting me for this wonderful conference. And I have to confess that I fall for the curse of the last speaker, which is to try to modify a lot of my talk just to try to see if it fits to the general topic, right? And hopefully I didn't mess up too much. And so I was saying that I'm interested in behavior in general, and I'm interested in how actually the behavior is originated at several scales, right? So at the evolutionary scale, for developmental scale, and maybe something that we could call like a real-time scale. Right? And the one behavior that I'm especially interested in is vocal communication. Right? And one of the reasons that I'm really interested in vocal communication is that it's sorts of universal behavior, at least among vertebrates. So obviously humans, mammals vocalize and vocal communicate. Birds also vocal communicate, obviously, and amphibians also vocal communicate, and even fish also they vocal communicate. Most fish they vocal communicate. Right? So in this sense, it's really an interesting behavior to do comparative work. But also, I would say that an interesting aspect about vocal communication that is quite easy to record, right? So it's not something that usually is one-dimensional, right? At least from the production side. So you can just have a recorder and start recording the acoustics and try to understand what's going on. In my case, yeah, I have a little bit of this anthropocentric bias, and I usually try to compare with what's happening in humans, right? And I think that there is some legitimate reason for that, right? Especially because we are humans, and obviously we understand our own umwelt. And when we think about humans, I would say that it's kind of natural to consider the primate family and try to understand better what's going on there. And today, I will talk about one specific species of primates that is marmoset. Right? So maybe here, most of you know marmosets, but marmosets are very tiny primates. So they weigh between 300 to 400 grams on average. They are natural from Brazil, actually. Here, and they're actually natural from the northeast of Brazil. But as you can see here in this map, they already migrated in pretty much the entire coast of Brazil. So now they are considered a pest. So pretty much they invaded the entire coast of Brazil and they migrated up to 5,000 kilometers. So it's kind of interesting that there is a primate species that in few decades actually migrates to the entire coast of Brazil. The other reason that I think it's marmosets interesting, especially for vocal communication, is because they are very vocal. So let me try to play here. It's a little bit loud, so just take care. And no. Good. Okay. Yeah, so this is a typical call. The first one, it's called the free call. Right? And the second one, the Twitter call. This is a typical type of interaction that they do in the wild. And actually, if you go to the northeast of Brazil, maybe not only northeast, even in Rio for sure, you will see marmosets everywhere. And another reason that always marmosets are interesting, as I said, they're primates, so they have a primate typical brain, right? So, for example, here is just a simple measure of the, let's say, brain complexity. So here it's a data set from Finlay and Brodsky. What you see is that when you measure the cortical areas, right, and plot against, for example, the number of cortical areas, and you compare, for example, the similar size rodents, you can see that the marmosets have a much larger number of cortical areas, right? And actually, a number of cortical areas that is quite similar to a much larger type, much larger primates, right? Also, one aspect that I think it's interesting with the marmosets is that they have some convergence aspect with humans. Right? So one interesting aspect is the cooperative breeding. Right? Very few primate species are cooperative breeder. And when I say cooperative breeder, it means that not only the mother but also the father and also the siblings, and even sometimes strangers in the group, they take care of the babies. So this is considered something that is very special among primates. We know only the characteristic family and the humans that have this type of behavior. Also now, we know that they have a vocal development that is plastic, where actually their vocal development depend on the parental feedback. And this also seems to be something special for marmosets and definitely for humans, as we know. So I said, I try to modify a little bit this talk such that try to approach some of the common themes that we discuss in these 2 days. So the first theme that definitely is the most common theme here is that we should understand how we get there. And I think that we have to understand how we get there, not only because time is an important parameter of any biological phenomena, but also, or maybe especially because I think in biology, a lot of things are contingent on the past, right? So a lot of things are really history dependent. So for us to understand what's going on, we should understand what's happened before. And then in the vocal behavior, then the question becomes, okay, when does the development of vocal behavior start? So maybe the usual way to think is that maybe it starts at birth, right? Because this is the first time that you hear any baby, or any human vocalizing. Right? But then when you start to think a little bit more, you say, okay, that's interesting, but maybe something should be happening before so that the baby can actually vocalize as soon as they are born. Right? So when you think like this, essentially you will start to look maybe inside the uterus and maybe try to wonder what's happening there. Right? Most probably there is no vocalization there. Right? Their lung is filled with water. Right? So probably they are not producing vocalization, but still they can move their mouth. Right? And we know actually from studies from the 70s and 80s that actually they produce vocal movement, mouth movements, sorry, that's quite similar to cries, right? Baby cries. And we know even that there is some sequence of jaw and mouth movements that are characteristic in human babies. We know that first there is an opening, and afterward they start to be able to close, and finally they will be able to move the tongue, right? This is quite classical, but there are very few studies that follow up this kind of evidence, especially there was no study doing longitudinal study. So we said, okay, marmosets vocalize a lot, right? They have a lot of homology and convergence with humans. So maybe it's an interesting species to try to understand what's going on in the womb. Okay? And that's what we did. So essentially, this is an ultrasound image of marmosets. So here, you will see the mouth, okay? It's a little bit creepy, but you see that there is an eye here, okay? The nose here is the mouth here, okay? And let's see if I can play. Okay, so you can see, I think that it's pretty clear that you can see the mouth movement, right? Yeah, some people think that is cute, some people think that is creepy, I don't know. Anyway, so now having this image, what we can do is actually to measure, for example, okay, the distance between the jaws, okay, and then try to figure out what is type of mouth movement, type of mouth movement they are doing, okay? And then what we can do then is to compare those kinds of mouth movements with the mouth movement that we see in the first day of their life after they are born, okay? And then the idea is to see if we observe mouth movements that's in the womb that are quite similar to mouth movement that we see while the marmoset is vocalizing, right? The baby marmoset is vocalizing, okay? So this was our first thing that we did. And what we see is that indeed, there is a correlation. So this is the gestational age. So this is the marmoset has a gestational age around 145 days on average. So you see that actually there is a small correlation between the gestational day and the distance, similarity distance. So smaller means that they are closer, right? But then you can say, oh, but Daniel, this is quite small, and I agree. But you can think that most probably they are not doing only mouth movements related to vocalization in the womb, right? So probably they are making mouth movement that is related to chewing, right? Suckling, and so on. So then the next idea is to try to maybe select some type of mouth movements that if they satisfy those criteria, they should be related to vocalization. Okay. Sorry, I forgot there's one here slide where you can see the comparison. And that's what we did. So essentially now we went back to the mouth movement that we saw in the womb, but we only calculated the percentage of those mouth movements that if they satisfy those characteristics, they should be related to the vocalization. And what were those characteristics? First was the length, because we knew that we measure all sorts of mouth movements after they were born. And we figured out that if the duration of mouth movements was longer than, for example, 6 seconds, right, they have a pretty much zero probability that they were not related to vocalization, right, for example. Right? And we also measure, for example, the frequency of the movement, and also that was characteristic for vocalization. And then when we went back in the uterus and we checked the percentage of the movement that satisfied those criteria, we clearly see that there is an increase and now a much stronger correlation between those mouth movements in the womb and the mouth movement that we saw after they were born. Okay, but then you can say, okay, but maybe this is something very general. Maybe any other type of mouth movement also is correlated, right? So we use as a control actually a chewing mouth movement, and then we saw that the chewing mouth movements actually we didn't observe any type of correlation. Right? Just what does it mean? Most probably it means that the mouth movements, well, that's a hypothesis because I don't have the data set, but the possibility is that the mouth movement related to chewing probably develop even earlier than what we observe for vocalization. Okay. Okay, so now the baby is born. Right? What they do? They cry. Yeah? They will produce some protophone. Well, they start to produce some sounds. But then soon after, well, maybe after 6 months, 9 months, they will start to produce adult-like sounds, right? And maybe after a year or so on, they will start even to have some conversation, maybe even start to argue with you, right? So it's quite remarkable type of development the human showed. And those kind of remarkable changes are not unique to humans, right? Maybe in a different extent, but definitely we know that songbirds show some remarkable development in their vocalization that is socially driven. And also we know that the bats have a clear vocal learning. But what's happened in the primate case? In the primate case, I would say that the literature for a long time assumed that there was no remarkable vocal development in non-human primates. So we wanted to recheck or revisit this hypothesis now on marmosets. And I think the marmoset is a really interesting model system to study development because they have, although they are primates, right, they have a much faster developmental time. Right? You can think roughly that the marmosets are 12 times faster. So the first 3 years of a human baby would be around 3 months in the marmoset's time, okay, developmental time. And then the experiments that we did, the first one was not even experiment. It's a really simple thing. We just put the baby, well, only for 5 minutes. We just separated for 5 minutes from the parent. We just recorded their vocalization, okay, pretty much every day. And what we got these kinds of spectrograms. So this is the spectrogram for the first day after they were born. And this is after 2 months. Right? You can already see that it's quite different. Let me see if I can play here. So this is the first day. So this is a kind of, let's say, immature feet kind of thing. There's a little bit of cry and this kind of thing. So let's go to the after 2 months. Okay, so this is after 2 months. So this is in the same context when they are alone. Okay, they definitely there is a change. Okay. And obviously, we should quantify this, right? Obviously, we can see the spectrum, we should quantify, and we measure several acoustic features. And then when we see through development, we see that, okay, there is definitely some change in these acoustic features, right? We can see in this way, or we can see how the songbird people like to see in this two-dimensional kind of feature. You can see that actually In the beginning, there is a kind of, the clusters are not very clear, but after 2 weeks, start to become clearer. But then finally, you kind of converge to a single one, at least in this social context. But obviously, this you could still argue that, okay, maybe it's related to growth, maybe other type of biomechanical factors. So then the question is really if these kinds of change are experience dependent or not, right? And for this experiment, I was really inspired by this paper by Michael Goldstein, King, and West, where they showed that in human babies, right, if the parents, right, respond contingent to a call, that's make the infant's calls more mature. Right? So maybe I thought maybe something very similar in marmoset could be happening. Right? So then we set up the following experiment. We just put the baby here, okay, and the adult parent here, right? And now we just recorded for 10 minutes, okay. And then there is a curtain here, okay, just simulating when they are far from each other, they cannot see each other, and we recorded their vocal interaction. And what we observe, right? This is one example, just to illustrate what just to make a point about the change in their vocalization. You see that in the very beginning, we see very few fees, okay? But there is a sudden change where there's an increase in the fee calls, right? Which fee calls is the fee type of call, right? And then there is also in the beginning, there is a lot of cries, okay? But then pretty much at the similar time points, there is a decrease on this price, right? So then it's kind of natural for us to consider this index, where is we take the difference in the number of fees and price and divide it by the total number of fee and price. Right? Why this index is kind of makes sense, because if it's negative, it means that you have more price. If it's positive, it means that there are more fee. Right? So, and then we can use actually this transition time, the zero crossing day, as a transition time for development. And now the hypothesis is the following, right? Because if we want to see if the interaction with the parents make any difference. Right? And to test this, what we did was to measure the amount, the probability that the frequency at which the parents responded to the infant's calls. And then we correlated with this zero crossing day. Okay? And what we see this, essentially, if the parental contingent is higher, which means if the parents responded more frequently, we see clearly that this speed up the development, okay? But then you can say, okay, this is interesting, but maybe it's not that the parental feedback that is relevant, but maybe just the infants that vocalize faster make the parents to respond faster. So maybe the causality is going the opposite way. Okay? Fair enough. So what we have to do is a manipulation experiment. So how we did that, essentially, we took all the sets of babies now, and we did a playback experiment where so this is one interesting aspect of the marmosets is that the marmosets usually give birth to twins. So actually we can use that. So one of them we can use as a control in our experiments always. So what we did was actually one of the babies, we give a higher contingent playback. So in 100% of times that the infants produce mature call, so fee type of call, the playback responded back. Right? But for the other twin, we only responded in 10% of time, okay? And then what we observe that the ones that received low contingents actually had the development that was much slower, almost constant here, compared to the one that received a higher contingents. Okay, this is one of the acoustic measures. So it seems that the marmosets change their vocalization, and this change is driven by the parental interaction. Okay, so the second theme that I observed here in the discussion was that we should understand the constraints of variation, no? In development. And the problem with that is that development obviously includes a lot of factors, right? So it's obvious, the vocal tract, for example, in vocalization case, the vocal tract is changing, the muscle size, muscle strength is changing, the brain is changing, even the social interaction is changing. And all those kind of things has a feedback, well, which make really difficult for you to understand what's going on, because you change one thing, you will change everything. Right? So then the question is, what we mean by understanding the constraints of development. So this goes a little bit of the yesterday panel discussion. What do we mean by understanding? I think that's something that comes close to understanding is the idea that was put forward by Waddington. I really like this idea of Waddington epigenetic landscape, where you have actually this landscape, right? And then you have this ball, and the ball rolls down, and then essentially just by chance, we'll go to one side or the other, right? So I think that this idea is really great, right? And there are some attempts to try to generalize this idea. And one of them is actually by George. And I was very inspired by this article, where one of the proposal is that if we are able to actually create those kind of landscape, maybe we can actually talk about homologs, for example, for behavior. Because now, if you have, for example, in George's case was for the neurodevelopment, but maybe if we have those kinds of landscape, and if we can compare two shapes, right, and the shapes are very similar, we can say that they're homolog, right? And so maybe it's a one way to quantify what homology of behavior means. So to do that, we have to first be able to calculate this landscape. And then I try to do that using the vocal behavioral data set that we already have. And the starting point was actually to try to understand the biomechanics of their vocalization. Right? So we know that pretty much the marmosets, as any other primate, has vocal apparatus where you have the lung, you have the vocal tract, and you have the upper vocal tract, the filter, what's going on here, down here, right? And then we can have a model for this biomechanical process, right? And we can try to see if this biomechanical model actually can reproduce what we see in real marmosets. So this is the cry, right, from the infant baby. This is what we got from the model, right? This is the type of call that we call subharmonic phi. You can see that it's quite similar. And the phi, right, it's pretty much identical, right? And we tested this model using excised larynx, and we compare, and actually the prediction matched pretty well with what we got in the excised larynx in marmoset. So now that we have this model, we can actually vary air pressure and vocal fold tension and predict, okay, what would be the energy efficiency of vocal production in marmosets for different parameters, okay? So just to simplify our talk here, so I would take just the diagonal here, right? Instead of considering the entire 2D, I would just consider the diagonal, and then you get this kind of image, right? And essentially, what we have is here is a cost function, where actually when the cost is low, it means that the animal, it's easy for the animal to produce a call with this parameter, okay? Where theta here represents the air pressure and the tension. Is this clear? I don't know if I went too far. Okay. And now using this cost function, okay, there is a standard way in physics to create the probability distribution, right, which is the maximal entropy principle. So now, knowing the cost function, we can tell you what is the probability that the marmoset would produce a call with this parameter, right, and so on and so forth. Okay. Obviously, there is one free parameter here that is called the temperature of the anthem. But what's interesting is that now we can think of development as a change in this cost function, okay? So let's say that actually maybe what's going on during development is that this cost function is changing, for example, the angle, right? Or maybe some other thing is going on, okay? Then what we did was to use this actually mathematical equation to our developmental data set, and what we got was actually this kind of landscape. So now we have a way to recover the landscape of marmoset vocal development. Right? And what's interesting is that we can make some predictions of what's going on because we know the meaning of each parameter. For example, if we change the vocal tract length, we have a prediction about what's going on the shape. Right? You can see that this thing now you create suddenly, actually you will create another type of vocalization here, right? If you change the muscle strength, also you will have some prediction of what's going on. Yeah, and the same for the neural chain and so on and so forth. Okay, and this is one video tried to show what kinds of what's the effect of knowing landscape in our understanding of the vocal development. So let's say this is the ball that in the Washington, right? And the position of the ball will tell us about the type of call. And here is the ratio between cry and feed, right? And you see that in the beginning, it's stuck here in the where there are more cries, okay? And here is the ratio, the index that I showed before. Yeah. And now you have higher chance to go to the other side just because actually tilted, right? The landscape tilted a little bit. And now, most of the time now, okay, the animal is stuck essentially to this thing. So this is, it's a one way to try to visualize the constraint, the multidimensional constraints that development has, and make it a little bit more intuitive to understand what's going on. Okay. But you can say, but still, I would like to see a more quantitative prediction of what's going on using this kind of framework. Huh? And that's fair enough. So then I come to the third point, I think the common theme of this conference, which is that we should try to explain how and why things change. Okay? So for this, I will start with noting what Rosa was telling us, that essentially in development, we see a lot of times we see stages. Right? So for example, when we look at the human, for example, vocal development, we see that in the beginning, they cry, they coo, but then suddenly they will be babbling. And then at some point, they will pretty much producing speech-like sounds, okay? And then the question is that how this transition happened. It is the case, right, that this transition happened in a linear way, a monotonic linear way, or maybe it's happened in a jumpy way, right? As pretty much Rosa was suggesting for us, right? In her example. And why this matter? I think that it matters because the difference between something that is go linear to something that has jumped tell us a lot about the mechanisms underlying this chain. Right? Just to give a further example of this point, I want to discuss a little bit the controversy that exists in evolution. Right? I think maybe some of you know that there is this discussion about gradualism and punctate equilibrium, and it's exactly this hypothesis, this discussion, if the evolution is something that goes in change linearly, like in this case, or it's something that jumps when you can see jumps, like in punctate equilibrium. Okay. So now the question is, what's happened in the vocal development? Maybe we can use the similar kind of, so we can actually pose the similar kind of question for development. And for this, we use data sets from 3 different animals, from the marmosets, from humans, and from bats. Okay. And just to try to make the model more concrete, I will make some explanation here. I hope that you bear a little bit with me. So it's exactly the similar idea with the landscape idea, but now we start with the probability distribution of calls. Right? So once we record the calls, we know that they have different distribution for acoustic features. Let's say duration here, right? And let's say that you had the duration had this kind of probability distribution. If you take pretty much the log of the inverse of this probability distribution, what you get is the cost, right? So whenever the cost is lower, it means that you have higher probability to have the call, exactly as in the previous landscape model. And what's nice with the cost is that now you can discuss, okay, maybe the cost is higher here because maybe there is a lack of, for example, energetic resource, right? Maybe it's more difficult for the animal to produce those kinds of calls because the cost is higher. Right? And when you go lower here, why the animal won't produce calls with this parameter, with this duration, let's say, okay? Maybe because if it's too short, right, there is no communicative meaning, so there is no information for the animal. It's okay? It's clear? Yeah. Then when we think about changing, okay, in the acoustic parameter, so here I'm calling something that's called principal acoustic component. You can think it as a principal component of the acoustic features, okay? And let's say that you see a change in them. A change in the acoustic component means a change in the cost, right? The position of the cost, okay? So it's shifting from something that was here, which means that the cost was here, is now shifting to the right. And now you can ask if this change in the cost happen linearly, okay? Or maybe it's happened in this kind of fashion where there is asymptote, right? This is very typical of body growth, for example, right? Or actually what's happening is that there is a balance between these two possible wells, right? Two components. And whenever it starts, let's say that it starts with this darker red, okay, cost, which means that it will be stuck here. And then this cost will be changing up, and then this side of the cost will going down. Right? So you can, let's say that you go to this lighter red. So what's happening here? It's happening that until this cost is lower than the cost in the right side, the animal will be producing this type of vocalization. Right? But once actually the height changes, right? Suddenly there will be this quick change that will go to another stage of vocalization. So this is a way to model actually this kind of sigmoid shaped vocal development. So and now the question is, which one fits better to our data set? And obviously, that's the point is that you can see, maybe you don't even need to fit the model here. You can clearly see that there is a jump in the acoustics, for example, in marmosets, in bats and humans. Right? But what's really interesting here is that this model, right, that we fit using the data set only from the beginning and end, actually predicts pretty well even the transition dates, right? So this is in the black, you have the transition date for the experimental data set, and in the red, what is predicted by the model, right? Mm-hmm. Well, okay, so this is fine, but then you can say, okay, this is just a sigmoid, right? There's nothing interesting. And that's also fair. But then I think that what's interesting with this model is now we have interpretation that is two costs. It's a balance between two costs. And then the natural question is, what are those two costs? Okay. And the one hypothesis is that, which is, I think it's reasonable for vocal communication, is that there is a balance between the metabolic energy available to produce a call, right? Because produce a lot of call is quite costly, but also there is a balance on the information efficiency. Right? So you want actually your vocalization to do something with the listener, okay? So if that's the case, what we predict is that in the very beginning, right, the cost would be one of the costs, sorry, would be related to the energy efficiency of vocalization. And the prediction is actually that in the beginning, the energy efficiency would be really high, but with age will go down. Okay. So how we met, and then what we did, we just to try to see if this is reasonable, we measure actually the energy efficiency of vocalization. And the way that we did this was to use heart rate, right, to measure the basal metabolic rate. And then we took the number of calls that the animal produced and divided by the heart rate, right? And that gives this kind of shape where in actually the younger ones, right, has a much higher energy efficient than compared to the older ones. Okay? On the other side, if the other cost is really related to the information efficiency, what we would expect is that the immature marmoset would be really inefficient from the information point of view, but would be efficient when it become older, it will become more efficient, right? And then we can measure that also, right? And the way that we measure was to use measure of information transmission, right? And essentially you can think that is if the infant vocalization is able to change the pattern of parental vocalization, it means that the information was efficient, right? If the infant produced a vocalization, but the parents didn't change their vocal pattern, it means that it was inefficient, right? So that's pretty much what we are measuring. And what we see is in the very young marmosets, the information transmission is inefficient, but when they become older, they become efficient. Okay. But again, it's just correlational data set because we just showed that, okay, the direction of the curves are similar, but now we want to use this idea to make some predictions. Right? And the first prediction is actually to try to figure out what's going on in this experiment that I showed before, right, with the playback in development. And what's going on in this playback experiment is that one of the marmosets is receiving playback every single time that they vocalize, which means that in this condition, the infants were highly information efficient, right? Because they are causing, let's say, the virtual parents are responding back 100% time. The other twins, actually, they were highly information inefficient, right? And then here's the prediction, right? So let's say that you have this energy efficiency curve, well, that should be similar to the both. But what's the difference between these two twins is that one of them are the one that actually received the high contingent playback are information efficient, and the other one is information inefficient, right? And what our prediction is that the one that received a high contingent would shift faster, right, before than the one that has a lower contingent. And we test it, and that's exactly what we see, right? So in the red is the one that have the high contingents, and the yellow is the one in the low contingents. You clearly see that the transition days are shifted because of this manipulation, okay? Now, what we want to manipulate is the energy efficiency, right? And then the question, how we can manipulate energy efficiency? One way to do that is actually to use heliox, right? Heliox is actually the mixture of helium and the 20% oxygen. So pretty much, from the respiratory point of view, there is no problem for the marmoset, but the air become much lighter, right? So then this make actually much easier, let's say the lung, right? It's much easier for the lung to move, and that's exactly what's happened. And what our prediction is the following, right? The one that received was in the heliox, the infant that was in heliox, right, become more energy efficient. So this curve went up, right? And the one that was in the normal air was more energy inefficient. So then this is interesting, that the prediction is that the one where the energy was more efficient, the transition they should shift to the right, right? Should like slow down, right? And that's what exactly happened also, right? So here in the blue was the one that received the air, and here in the green, you clearly see that the transition that was shifted to the right, right? So this shows that, okay, it's a kind of evidence that to show that maybe really the energy and information are two factors that are modulating the speed of transition in the vocal development in the marmoset. Okay, so finally, the common theme is that, and that the one that I really like is that maybe brain areas that are conserved, although they are anatomically conserved, maybe they have dynamics that are not necessarily conserved, right? And that actually comes from, so I was very motivated or influenced by this paper by Xiao and Finlay, Barbara Finlay, called the Thinking Outside the Cortex, right? And then what she says is that the alteration of the organization of social and motivational anatomical circuitry must have been an essential step in the evolution of human language, right? And this is kind of interesting because what she's alluring is to the fact that maybe we've been too much like cortex-centric and actually very lateral cortex-centric, right? So it may be more medial and subcortical areas could be relevant actually for the evolution of language, right? So, and this kind of fits to at least to the non-human primate literature. We know from very classical Jorgen study, right? Several dozens of studies from Jorgen that show clearly that there is this medial sagittal areas that are highly relevant for vocal production, yeah, and yeah, for vocal production in general. And also we know that the same sagittal medial regions include the very relevant social behavior networks, right? Which include the anterior hypothalamus, preoptic areas, right? Ventromedial hypothalamus, lateral septum, PAG, and extended amygdala, which are the areas that we know that they participate in pretty much every single social behavior that we know, right? And also from the Jürgen study, we know that the ACC, right, the frontal-orbital cortex should be relevant also for vocalization, okay? And now our task was, okay, we want to study these areas, but these are medial and kind of deep areas, and how we can record all of them simultaneously. Because we really wanted to see what's going on in all these areas. And the one way to do this was actually to use a technology called the functional ultrasound imaging, right? So this functional ultrasound imaging, essentially it's an ultrasound technology, right? And as any ultrasound technology, it's measure the scattering of the matter. So essentially it sends a probe sound, right? And then it's what comes back is directly related to the density of the substance that is there. Okay. So knowing actually the speed of the sound, we know actually the distance also. And because we have this neurovascular coupling, we know that whenever it's increase actually the neuroactivity, and that's cause an increase in the vasculature, that should increase actually the scattering. And that will create an increase in the signal in the functional ultrasound, okay? So we use this technology, and the one really nice aspect of this technology is that it's really light. So it allows us to record this big chunk of the brain in animal that is still behaving, right? And if you want to study vocalization, that's really crucial. And also because we are using marmosets, which are primates, right, we really want these kinds of technology to allow us to record for a long time. In our case, we were able to record for more than a year in every single marmoset. Right? Just to discuss a little bit about the resolution. So in our case, the temporal resolution were 500 milliseconds. Some people use 100 milliseconds, but we choose 500 milliseconds because the signal to noise ratio is much better. So in the x-axis, which is in this axis, so it's in the, when you think in the sagittal plane of marmosets in this direction, right? It's 150, 125 micron, right? The depth is around 130 micron, and the thickness, it depends a little bit on the distance from the probe because it works like a lens. But on average is around 400 micrometers, and in the focal region is around 200 micrometers. Now, using this function ultrasound, we ask if this social vocal network is really activated by vocalization. As I said, the most of literature focused on the production side. Now, we wanted to know if these areas were also activated during listening, right, during perception. And then we did a very simple design experiment where we put a marmoset here, we put a curtain, and we did a playback. Okay? And using a playback experiment, we got this kind of signal. So this is a single trial, okay, from the anterior cingulate cortex. And what you can see is that the signal to noise ratio is pretty good. Right? So this every single trial, you can see actually that they go up and down. Right? And for the first thing is that we just wanted to know if this kind of the social vocal network is activated at all when they listen to when the marmoset is listening to vocalization, and you can see clearly that they are activated. Yeah? And obviously they kind of match to this social vocal network that Jürgen described from the production side. This is just a video just to show what kind of things we obtain with this functional ultrasound. Right? So this is a marmoset brain, okay? This is an average of 20 trials. And then what you will see is that it starts 9 seconds before the playback. And when the playback starts, you will see a blue color here, and then you will see the activation. Right? The activation will be active. The active areas are shown in orange here. So you clearly see that is activated and it is exactly corresponds to this kind of social vocal network. It's a very subcortical. Maybe there is a include the ACC also. Okay, but that's just show that okay, they respond to acoustic, right? But what is interesting about it? If it's really social, right, and it's not only acoustic, you would think that they should be modulated by the social aspect of vocalization. And what's really nice with the marmosets is that we know a lot about their vocalization, and we know that they produce calls, which is this one, that probably now you're tired of listening, which is they produce when they are far from each other and they cannot see each other. Okay? And they also produce this type of called trilphy, which is quite similar to phi. Right? There is a little bit of trill in the beginning, but it's quite similar to phi, but they only produce when they are close to each other and they can see each other. Okay? And they have also this tweeter that acoustically is very different from phi, but actually they use in the same context, social context as the phi. Oh. Okay, so this is really nice setup where actually you have two acoustic similar calls, but that are using in different social contexts, and two acoustic dissimilar calls that are using the same social context, right? And now what we want to know, okay, what's happened in their brain. Okay, so here you have the phi, and here have the true phi, and let's see the areas that where we see activation difference in the functional ultrasound. And you see that actually it's a lot of areas, right? Especially again, you know, not maybe coincidentally in the social vocal network. Right? And now we do a comparison between phi and Twitter, and this is what you get. Okay? Pretty much no difference, right? Essentially. And you can say, oh, maybe this is just because the peak is the same, you know, but maybe the dynamics for each, the neurodynamics for different calls would be different. So this is the when we compare the dynamics of the brain activity when the phi and the Twitter, you see that they are actually quite similar, but indeed for phi and true phi, they are quite dissimilar. So this really shows that actually the social vocal network, right, seems to be really relevant to figure out the social context in which the vocalization are producing. Now, again, you know, the question is that what happened during vocal production? And we already know that these areas are relevant for vocal production for Jorgen's study. But now we want to know if actually these areas are also relevant to differentiate between different type of vocalization when they're produced, right? And then to do that, what we did was actually to do a playback experiment where the marmoset was on the side, the speaker was in the other side, and we have actually interactive marmosets, right? I didn't talk so much, but we have a model of a marmoset's vocal production that can interact with the marmoset. And then we use this to probe, to make the marmosets to interact with the computer, right? And then they produce vocalization. And what we see is that indeed, there are several areas in the social vocal network, like ACC, posterior hypothalamus, where the lateral septum, where you see actually activity before the vocal onset. Okay, and these same areas, pretty much you see that again show difference for different type of vocalization. This is when we are comparing the phi call when we compare to the alarm calls. Okay. Finally, we wanted to see if actually this social vocal network also reflects temporal context. What do I mean by temporal context? It means that let's say that you are looking at two vocalizations, but one of the vocalization was externally driven. So it was a response to a vocalization from other marmosets, in this case, actually a playback, right? And we wanted to know if these areas also were relevant in this kind of context. And this, although they are producing exactly the same call, right? The only difference is they're listening to, in one case, it was driven by external factor. Right? And then when we see again in the functional ultrasound, we see that those are the areas that seems to differentiate if the temporal context, right? If there was another call or not, if it was a response or non-response to another call. Right? this is so just to say that this subcortical area seems to be doing something that is quite relevant for vocal communication, which is to distinguish social context and even temporal context, right, in a in a, in a communication context. Okay. So some take-home message just to finish. Pretty much what I want to say is that the behavior developed in highly nonlinear way. Okay, it could be that it's so nonlinear that things that are not vocalization, like just mouth movements, could be relevant for development. Yeah. Quantitative modeling of developmental constraints could give a framework for understanding animal behavior. At least I hope that that will help us to understand better animal behavior. And one thing that I think that is really interesting and standing, especially for people studying primate, is that we should definitely look at non-cortical areas that they could be really relevant for understand evolution of behavior, such as speech and maybe language. And with this, I would like to thank all my collaborators and the funding agents. Thank you so much.